First of all, I reiterate what Rochelle just said. Nothing I'm about to say constitutes medical advice. I'm not a physician. I am a researcher. And so we'll talk a lot about some tests and use of certain genes and information we glean from genes today. But again, none of that constitutes medical advice. So who am I? Why should you be here or care that I'm here? Perhaps you shouldn't. So I just a little background on me. I did my Ph.D. at the University of Ottawa from 2001 to 2006. September 11th, 2001 was actually my first day of graduate school, so it was a bit of an inauspicious start. I did postdoctoral training at the Princess Margaret Cancer Centre here in Toronto from 2007 to 2010. I then became the scientific director of the Canadian Prostate Cancer Genome Network, which you will hear a lot about today. And as a staff scientist here at the Princess Margaret Cancer Centre and assistant professor at the University of Toronto. Just recently, I've moved to Movember Canada as a director of implementation in cancer programs. And this is really about setting up some new initiatives that hopefully you'll be hearing about over the coming months, where Movember is really trying to broaden its position around directly funding prostate cancer research, but also survivorship programs and a lot of other related programs. So hopefully you'll be hearing more about that in the coming months. So here's what we're going to discuss today. And as Rochelle said, if you have any questions, please just put them in the chat. I'll be happy to get to them at the end. It's no problem at all. Take any and all questions. So we'll talk about the concept of heterogeneity and this thing called precision medicine or personalized medicine, and what it means, what it should mean, what it often does mean, why it often doesn't mean what you think it does, and some of the issues around the concept of disease heterogeneity, and how that relates to prostate cancer. What's the critical problem, or one of the critical problems with prostate cancer? We'll do a quick biology refresher for those who haven't seen kind of this stuff in a while. We'll kind of go over some of the basics that you'll need just to kind of understand some of the stuff I'll show later in the talk. We'll talk about how we use genetics and genomics to improve prostate cancer outcomes. I'll talk about some of the studies that we've been doing, again, over the last 10 years or so within the Canadian Prostate Cancer Genome Network. And then finally, end with a short discussion on how we actually move this from the research stage into the clinic and what's going on behind the scenes to actually push forward some of this research, because ultimately that's why we do this, to get these types of this type of work translated into the clinic. So we'll start at the beginning, which is always a good place to start. The problem of heterogeneity. So what do I mean by that? Well, consider the concept of overall life expectancy. So this numbers are a few years old, but in Canada, life expectancy at birth is somewhere around 82 years. It might be a little higher than that now, but generally speaking, it's around that level. And what does that mean? Well, it means that the average Canadian born at this time, 2018, could expect to live 81.95 years, which is interesting in and of itself. But what does that really mean? What that actually means is that the average person, 50% of the population, or what we call the median of the population, will live to be that old. It doesn't mean that everybody's going to live to be exactly 81.95 years old. There are, unfortunately, people who die in infancy. It's very, very small, but it does happen. Conversely, there are people who live to be 120 years old. Again, it's quite rare, but it does happen. And so life expectancy for the average man, I guess in 2021 would be about 82 years. This is a really useful thing when it comes to measuring the health of populations. So if you want to compare country A to country B, you can say, well, what's the average overall life expectancy? And you can say, well, this country is healthier than that on average. But it's very unhelpful for measuring the health of an individual. The way we think about it is, who is this average person? Who is this average man? And why should I care about him? It's great that populations are living longer, but at the end of the day, what we really care about is individuals. And so this is the problem of heterogeneity. When you assign an average to something, it really speaks to populations, not individuals. And this is really true of anything that we can measure. So I'm a huge baseball fan. This is a plot of the height in inches of every single person who's ever played in Major League Baseball. And so the red line is sort of the average. And you can see that sort of this big cluster of people right around 72, 73 inches. There are a few people who are quite a bit taller and a few people who are quite a bit shorter. One, in fact, who was quite a bit shorter. And, you know, this is two quite famous baseball players. For those who follow, this is Aaron Judge and Jose Altuve. Aaron Judge, I think, is 6'6", and Altuve is, I think, 5'4". And then way back when we had Eddie Gaedel, who was, I think, 3'10" or something like that. It was a bit of a publicity stunt, but he was the shortest person to ever play. The point being here is that anything we can measure and assign an average to has a range. It has heterogeneity. So we'll come back to how that applies to precision medicine. But first, I want to talk about what precision medicine actually is. The best example I think I can give of precision or personalized medicine is you hurt your arm. You know, you're riding your bike or whatever, you fall off, you hurt your arm. You go to the doctor, you get an x-ray, says you have a broken arm, you put a cast on the arm, and then 6 weeks later, you're a broken arm survivor. That's precision medicine. It is a person with a specific ailment. There's a specific test to see what's wrong, a specific treatment, and then at some point, someone go unmuted there, I think. And then at some point, that treatment leads to a positive result. What precision medicine sometimes is, however, is this. You hurt your arm, you go to the hospital, the doctor looks at you and says, well, we know that the chance of you breaking your right arm is 3 times higher based on all of this published data. Therefore, we think you've broken your right arm and we'll put a cast on your right arm. The only problem, of course, is that as you notice, this person has a left arm, their left arm is sore, and they put the cast on the right arm because the data suggests that most people have sore right arms instead of left arms, or broken right arms instead of left arms. That's often what precision medicine is. It's using population level data, as I was talking about with life expectancy or heights, to apply to individuals. And what we really want to get to the point of in the concept of personalized or precision medicine is giving the right treatment to the right person at the right time. So how does this relate to prostate cancer? This is a plot you may have seen before of the incidence rates and the mortality rates for prostate cancer for the past 40 years or so. And of course, we have this big spike in the late 1980s, early 1990s when the PSA test was introduced. And it's kind of declined over time as treatment's gotten better and prevention has happened and all kinds of things. The important thing to understand here is that the vast majority of people, even today, particularly in the PSA era, are diagnosed when the cancer is confined to the prostate gland. So this is localized prostate cancer. No evidence of spread outside of the gland. And the 5-year survival rates for localized prostate cancer, even high-grade localized prostate cancer, is nearly 100%, 99%. 50% of all of the disease that will go on to threaten a man's life, that is, become metastatic or spread outside of the prostate gland, is going to result from localized prostate cancer that recurs after treatment. The other 50% is men who present in the clinic with what's called de novo metastatic disease, or the disease has already escaped the prostate gland. That's only about 10% of all diagnoses, but it contributes to quite a bit of the overall burden of advanced disease. And we take the view that anybody who has a localized prostate cancer can potentially be cured of their disease, because the disease has not, as best we can tell, spread outside of the gland. And thereby, if we are able to effectively treat that disease at the right time and quickly enough with the appropriate amount of aggressive treatment if needed, then those cancers are potentially curable. This is a sort of a schematic that I just drew of a kind of a typical man's progression through prostate cancer. You guys are probably well aware of this, probably better than I am, in the context of if you measure PSA values over time. You're going along in your life, you go for an annual checkup, and they say, oh, your PSA is rising. Maybe you go back 6 months later, you get another test, and at some point it passes a threshold that's usually 4 nanograms per milliliter. And at that point, you have a biopsy perhaps, and there's cancer detected, you might have surgery or radiation, and the PSA levels drop down again, and PSA never rises again. That's a really good. I mean, if you're going to, if you have cancer, that's an excellent response. Occasionally, what will happen is that same thing in the early stage of the disease happens. PSA drops back down to baseline, and then you might get a little blip. That's what's called a biochemical relapse. More often than not, that's easily treatable with radiation or short courses of hormones, and the PSA drops back down and again stays down for the rest of the man's life. And those are what we would, at least clinically, call cure, right? So the man has been treated and the PSA is remaining low. There's no evidence of tumor growth. In essence, that is a successful treatment. Of course, on the other hand, in a relatively small proportion of men, about 20% of the time, particularly with higher grade disease, you have that same progression. You might have a biochemical relapse, and then PSA starts to rise again, and then you have to get into things like systemic androgen deprivation and chemotherapy and all of those things. And unfortunately, this is an incurable disease. The good news is about 80% of the men fall into the first category, and only about 20% of cases fall into the second category. So our goal here, insofar as the research that we do, is really distinguishing these guys in the 80% from these guys in the 20%. And it's not easy because you can't do it after the fact. You can't go, once a man has been diagnosed with aggressive prostate cancer and it's metastasized and all these things, you can't say at that point, well, you're going to have aggressive prostate cancer. We know that. We need to be able to predict it up here. And unfortunately, these cancers often look very similar. We have ways of doing that, of course, things like the Gleason score, which many of you will be familiar with, which is essentially a measure of how the cells look under the microscope, how organized the glands look. It's a score from 1 to 5, 5 being very disorganized and consequently a more aggressive disease, and 1 being very well organized and potentially being a more indolent disease or having a less aggressive outcomes. And that Gleason score, along with the size of the tumor and whether or not the tumor is bilateral or not, potentially some imaging, and PSA allows physicians to categorize the disease as either low, intermediate, or high risk. And that's low, intermediate, or high risk of metastasis and potentially even of prostate cancer specific death. So as I said, most of the cancers are diagnosed in this localized phase. They're triaged into these groups. But the thing is, even then, what we're really still doing is applying these kind of broad statements about, well, larger tumors tend to be more aggressive, or disorganized glands tend to be associated with more aggressive cancers. These are really, again, going back to this issue of heterogeneity, these are really about population level observations that we're trying to apply to individuals. And I'll give you an example of where that sort of falls down. This is some data from a very large clinical trial that was published a few years ago now in the New England Journal of Medicine, really the top tier journal. And this is called a Kaplan-Meier curve. Essentially what it is on the horizontal axis is years, time in years here, and on the vertical axis is the number of patients surviving. So of course at time 0, everyone's alive, and then at some point the people are either still alive or have died of prostate cancer. So this is a study that was looking just at men with low risk prostate cancer. So you can see that at 10 years, virtually everybody is still alive. And in fact, in the 1,643 men that were enrolled in this study that either got surgery, radiation, or active surveillance, there were only 17 deaths from prostate cancer. So a 1.03% mortality rate. That's great. And so the pro there is that the population rate is very low. It's only 1% of men. The con, of course, is that those men still died. And those are deaths, and those are families that have lost brothers and fathers and sons and uncles and all of this. And so the reality is we used a population level metric. We classified them as low risk, but they actually had disease that was going to threaten their life. And so our job is to figure out, how do these men, these 1% of men, or in other risk groups, it could be as high as 10 or 20% of men, how do they differ from the guys who survived for not just 10 years, but for 20, 30, 40 years past their treatment. The point I'm trying to make here with all of this is to say that there is no average prostate cancer, and that's true of all cancers. Just like there's no average person. That person doesn't really exist. We have to be able to consider each man as an individual, and identify the unique risk of aggressive disease in that individual man and treat that person, or not treat potentially, based on what we know about that person and that disease as an individual case. And the challenge is really trying to figure out how to do that. Our thinking is that, as I said, all clinically localized prostate cancer is potentially curable. And again, I introduced these categories of low, intermediate, and high risk. And this is some data, again, another Kaplan-Meier curve showing time in years. And here is this biochemical relapse. So this is a PSA rise after surgery or radiation. And when you classify these men as low, intermediate, or high, you can see that low risk men at 10 years, about 90%, 85% of them did not have a biochemical relapse. In the intermediate risk group, it's about 50%. And high risk, it's about 30%. But if you look deeper into this, there are low risk men who are failing their treatment. They're having a biochemical relapse within, say, 2.5 to 3 years. And we know that that's a surrogate for very aggressive disease. And those men really need to be treated like they had intermediate or even high risk prostate cancers. And at the other end of the curve, there are people with high risk prostate cancer who are surviving and not having any disease relapse within 10 years. And those men potentially could be spared from some of the more harmful side effects of aggressive treatment. And the fact is, these risk groups simply do not tell us all of the information that we need to know, because they are based on population level aggregates and averages, not individual tumors. So current prognostic factors do not accurately predict clinical outcome for individual men with localized prostate cancer. And that really is our challenge. So before we get into some of the data and some of the ways that we've approached this, I can tell you that the general approach has been to look into the DNA and the genetic information that is present in men with prostate cancer from birth, as well as men from prostate cancer in their actual tumors. And what do they look like, and how do they differ between men who have a more aggressive case versus men who have a less aggressive case, all other things being equal. So same Gleason score, same all of the other stuff, same PSA, and so forth. So before I get into that, I just wanted, for those who are not familiar with this, I just wanted to give a quick, what I said, sort of high school biology refresher. Of course, for DNA is a very simple molecule, actually. It's a double helix with a sugar phosphate backbone shown here in yellow. It has 4 letters or bases, as they're known: adenine, thymine, cytosine, and guanine, A, T, C, and G. They, just like our alphabet has 26 letters, the DNA alphabet has 4 letters, and the order of those letters really determines what an individual gene does. So there are 3 billion bases, 3 billion letters in the genome, which is the collection of all of the DNA in a cell. There are 2 copies of that per cell, so 6 billion letters, 2 times 3 billion. Every cell you have has 23 chromosomes from each parent. Those are shown here. So any given individual has what are called autosomes, 1 to 22, plus a sex chromosome, for men XY, for women XX. And what's interesting is that there are only about 23,000 genes in a human cell. And that those 23,000 genes do not span the entire 3 billion. They actually only consist of about 1 to 2% of all the letters. So much of the genome is not actually a gene that codes for something, that does something in a cell. It's structural, or we don't know what it does, frankly, a lot of the genome. But that becomes important when you start talking about some of the mutations that we'll talk about later. Another important thing to remember is that A always pairs with T. So here you can see in the middle, between the backbones, you see the magenta next to the green. So these actually form a chemical bond. So A always pairs with T. G always pairs with C. And as I said, the sequence of the A, T, C's, and G's determines the function of a gene, just like the sequence of letters in the alphabet determine what the word is. And the order is not just the contents in terms of like ATG is not the same as CTG. The order of ATG, for example, is not the same as TGA if you mix those up. So both the which base you have as well as the order in which they come is very important for telling a gene what it's supposed to do. The single most important thing a cell has to do in its life is to accurately copy this. You have about 700 trillion cells in your body. Almost all of them are dividing, and every time they divide, they have to copy that whole genome, and it has to be done accurately. Because if it's not done accurately, what will happen is there will be changes in these base sequences, and that will change what the gene actually codes for. Within the central dogma, as it's known, of molecular biology, which is that the DNA codes for RNA. RNA has become really, really famous in the last 18 months or so from all the COVID vaccines. And then RNA codes for protein. And protein is really the thing that does the work in the cells in terms of causing cells to grow, to divide, to metabolize sugars and other nutrients, the structure of the cell. Whether the cell is a liver cell or a skin cell or a brain cell is determined by proteins. But ultimately, it all goes back to the sequence of these bases in the DNA. And of course, there are many traits that are inherited because of differences we have between all of us in our DNA. Identical twins are identical because they share an identical genome. They inherit the exact same DNA sequence from their parents. And then there are other features like hair shape, whether you can or cannot grow a beard. Some of us cannot, not saying who, perhaps me. The shape of your eyebrows, whether you're balding, hair color, all of these things are affected by individual genes or sets of genes, and they're inherited. You inherit differences from your parents versus your brother or versus someone that's unrelated to you. You'll have different sequences in these genes that govern what the feature will be in the end. But remember that other things can be inherited too. And so if you think of cancer, what cancer really is a disease of this genome. It's a disease of mutations in this genome, or changes in either the sequence or the structure of the genome that leads to inappropriate cell growth, cell death, cell survival, all sorts of things that happen because the genome isn't being regulated properly. And like any other trait, cancer risk can be inherited. So the most famous example of this, I think, is probably the BRCA1 and BRCA2 genes, which were originally discovered and named because of their association with breast cancer that runs in families, so familial breast cancers. This is a typical pedigree where we have circles are women and squares are men. You can see a woman here in the top generation had breast cancer. Her sister had ovarian cancer. Their brother did not have cancer, but he passed on the mutation in one of these two genes to his children. So his son had prostate cancer, his daughter had breast and ovarian cancer, and so forth. And you can go down. This is typically what it looks like. Not everybody will get cancer. Here's a son who didn't get prostate cancer. There's some other children that did not. But what's happened here is that the risk of the disease has gone up significantly. And so this inherited risk can actually account for about 5 to 10% of all prostate cancers, which of course leads to the question of what's causing the remaining 90%. And that's when we have to talk about things of the difference between mutations that you inherit from your parents, germline mutations, which are present in all cells of the body, versus what's known as a somatic mutation. So somatic mutations, soma is the Latin word for body. These mutations or changes in the DNA occur in individual cells, and they're caused by external factors. And these are the types of things you think about when you think about what causes cancer. So things like tobacco smoke, UV light, environmental chemicals, and even random copying errors. The cell, as I said, its important job is to make sure that the genome is copied accurately. Well, that doesn't always happen. There can be random errors that happen. And so assuming your cells are exposed to one of these chemicals, you get a mutation. I've indicated here by the X. That's only present in that individual cell. What's the effect on the cell? Well, most of the time, the effect is absolutely nothing because, as I said, genes are only 1% of the genome. Most of the time, these mutations happen in areas of the genome that have absolutely no role to play. They don't regulate anything. We don't know what they do, but the effect on the cell is virtually nothing. Occasionally, you will get mutations that are in genes, and occasionally it will occur in a gene that leads to, let's say, an increase in cell growth, okay? I'm saying 0.001%, but it's actually probably much, much lower than that. What's the effect on the person? Well, most of the time, it's nothing because the cell has evolved many ways in which to deal with these types of things, and changes in the genome can be fixed and they're not passed on, and the cell stops growing until it can fix the error. But occasionally, again, let's say 0.001%, but it's probably even less than that, that can result in uncontrolled cell growth, which is cancer, right? And so we have mutations that essentially are doing nothing, and we call these passenger mutations. And conversely, we have mutations that are promoting the growth of cancer, and we call these driver mutations. And this will become important when we start talking about some of the changes that we've observed that are associated with more aggressive disease. So what types of mutations can you get? Well, you can get changes in the number of copies of a gene you have. So as I said, you inherit for every region of your genome, every gene, you have 2 copies, one from mom and one from dad, noted by yellow and red here. Through time, in individual cells, you can lose one of those copies, which this is a copy number loss. And these are what's known, the genes that are affected like this are what's known as tumor suppressor genes. And you can also gain copies. So in this case, this person has, in this individual cell, has gained 2 additional copies of the yellow gene, or the yellow chromosome, or whatever you want to say. It can be quite large or quite small. And this is a somatic copy number gain, and this happens in genes that are called oncogenes, and they actually promote cell growth and cell survival. And I liken this to when you're driving your car. You have a gas pedal and you have a brake pedal. You want to make that car go, you have to do 2 things. You have to put your foot on the gas, so you have to amplify or increase the pressure on the car to go forward. That's akin to an oncogene, but you also have to take your foot off the brake, and that is you lose these genes that are involved in actually stopping cell growth. And once you do both of those things, the car can go. So those are copy number aberrations, or CNAs. You can have changes in the DNA code itself. So these are single nucleotide variants, or SNVs, sometimes known as point mutations, and they are literally changing one or a few letters in the code. So if this is the DNA code, DNA is a double helix, of course. So it has, every time you see a T, you will also see an A on the opposite side. So for example, a TA changes to a GC. Most of the time, that's not going to do anything. But sometimes if that is in the wrong place, it again can cause the cell to either become hyperactive and grow and survive when it shouldn't, or it can remove the ability of the cell to stop inappropriate growth or survival. Again, akin to the sort of gas brake analogy in your car. And then you have things like reordering of the DNA. So you can actually move bits of the genome around from one place to another. And this is usually involved in activating genes. And what will happen sometimes, and there are famous examples of this in certain cancers, where you have one gene that's moved next to another gene that's always on in a particular cell, and that gene will just be turned on all the time, all the time, all the time, and it will promote the growth of that cell. So this is in essence more like just stepping really hard on the gas pedal in an individual cell, and you will get that causing the car to go or cancer to develop. So some of these mutations, as I said, lead to the development of cancer. Some mutations can lead to more aggressive cancer, and some mutations can do both. And our mission as researchers, as translational researchers and clinical researchers, is to identify those mutations, both somatic, the ones that occur during your life, as well as the germline that you inherit from your parents that increase an individual man's risk of both developing cancer, but also having aggressive prostate cancer. And then ultimately to develop tests to identify who these men are, who is at high risk, or in fact at low risk of developing potentially life-threatening disease. So the question is, how do we find this? Because we have a 3 billion letter code. So how do we actually go about finding these mutations? So in the distant past, 2004, this is the speed at which some of these things move. We had what was called Sanger sequencing. And this was, in essence, When I was a graduate student, you would literally hold up a piece of X-ray film, and you'd do some chemistry to DNA that you had extracted from a cell, and you'd hold up a film, and you'd see what it looks like here within these sort of light and dark regions of this, and you could actually read down the film or up the film to get the sequence of these letters, these bases, A, T, C's, and G's. The chemistry behind this is not relevant here, but we had to do this manually and tick off each one. And of course, there's errors associated with that, and it takes forever. When I was a graduate student, we had this on computers. We could do this automated with computers, where instead of a band on a piece of film, you'd actually get a peak that was one color or another based on what the actual letter was, and the computer would read this off, which is great. But even then, we're talking about 10 letters per minute that could be read, even by a computer. And if you do the math, that works out to sequence an entire 3 billion base pair or letter genome. It takes about 570 years, which is obviously too long. So 570 years for a single human genome on a single sequencer. So what's the solution to this? Well, there are really two. Either you get lots of machines. So if you had 570 machines, you could do it all in 1 year. Or you shrink the size of the machine onto the size of a computer chip. You make each chip do the work of 1,000 machines, and then you can really step things up. And in fact, that's what's happened in the last 10 to 15 years, is we have a really paradigm shift in the technology to actually read and sequence DNA. And so by 2008, we have the first publications of the complete genomes that are sequenced by what's known as massively parallel or next generation sequencing in April of 2008. For those who are familiar with this, is the genome of James Watson, who was one of the discoverers of the structure of DNA. That was published in 2008. And then by fall of that year, November of that year, we have the very first cancer genome that was published. So actually not just sequencing the white blood cells from the person, but actually sequencing the cells that are cancerous themselves. So this is a sea change in our ability to go from taking hundreds of millions of dollars and hundreds of thousands of people working to sequence single genomes to being able to do individual genomes in a matter of weeks. And so like anything else that involves huge amounts of data, you need huge amounts of computing power. This is a picture of the Supercomputer Cluster we have here at the University of Toronto, which has 80,000 cores compared to the 8 that I'm on, the laptop I'm on right now. It's actually the 113th fastest cluster in the world, computer cluster in the world. And whereas I said earlier, if you had to read these off at 10 bases per minute, it would take 570 years. We can now do this in about 7 days to generate the data, and we generate a whole lot more data. And it takes about 7 to 10 days to do the primary analysis, actually do the computational work associated with that. And the main advantage here is not necessarily that these computers are any faster, but the fact that we can use 80,000 of them at once. So to do 100 genomes, instead of taking 1,000 days, might take 100 days, which is a much more manageable amount of time to work on. And of course, that has resulted in a massive reduction in the cost, from roughly $100 million to do a single human genome, to sequence a single human genome in 2001, to roughly $1,000 today. And so that allows access to researchers in ways that could not have been done before. So how do we use this? Let's get back to the question of how we actually use this technology and the knowledge we have about DNA and mutations to actually make differences and learn something about prostate cancer. So I've entitled this section "Going Forward by Going Backward," and I think you'll see why in a second. The situation is that we have groups of people, the blue men here. All of these blue men have prostate cancer. So what do we want to know? We want to know which of these men actually have aggressive prostate cancer. Now, unfortunately, they all look the same. They're all blue. So what do? We take their tumors, we sequence the DNA in each of their tumors, and we can generate profiles. And so it doesn't matter what the individual data points are here, but suffice to say each of these bars represents a different type of mutation here on the top, in the middle, and the bottom, and each one of the columns here is an individual person. We sequence each of these tumors, and then we stratify the men by how they did in the clinic. who had aggressive cancer, who had indolent cancers. So we reveal that information because we've collected these people what's known as retrospectively. So we've gone and found men who'd had treatment, say 10 years ago. We know how they performed, what happened in the clinic to them. Did they relapse? Did they not relapse? And so we can reveal that information. We now know that these 3 men are actually red. They had aggressive disease. And then we look for which mutations are actually associated that occur more frequently in these men, these 3 men, or might be 300 men versus these other 7 men, or it could be 700 men. And so by doing that, we can hopefully tease out the mutations that are common to these guys and not these guys. And that may give us some clues as to why these guys had more aggressive treatment, despite the fact that they looked exactly the same as everyone else. And that's in fact what we did within the Canadian Prostate Cancer Genome Network. So we looked at 500 patients, all of whom had intermediate risk prostate cancer. So they looked identical under the microscope. They had identical PSA values within a very narrow range, identical size tumors. But we know from historical data that 20 to 30% of these men are at risk of developing metastases after radiation or surgery. So we had 150 men who had radiation, 350 men who had surgery. All of them had at least 5 years of post-treatment follow-up. So what I mean by that is they were treated at least 5 years in the past. And so we knew what had happened to them over the course of that 5 years. We knew what their clinical course was, so to speak. We got the tumors from all of these, and we sequenced the genomes of all of these tumors to see what the mutations actually looked like. And we matched those to patient outcomes. And actually, by the time we had sequenced all of them, we had not just 5 years of follow-up, we had almost 10 years of follow-up on most of these men. I think at this point, we have almost 20 on some of the men. And so, as I said earlier, what we want to do is identify the mutations that predict poor outcomes. And really what that is developing what's known as a biomarker, right? It's something that tells us, something biological that tells us about something that's happening inside in the clinic. This is the team of individuals. These are obviously very large initiatives. This is just a small subsection of the team that led this study. So what did we learn? Well, we learned that prostate cancer, as I started off the talk, is very heterogeneous. So what you're looking at here again is a plot of the mutations that we found in each of the tumors. So each of these bars represents an individual man's tumor. And this is the number of point mutations, or those base changes where we saw an AT switch to a CG, for example. And the axis here represents the number of those that we found in their tumor. This is the rearrangements where you have DNA moving from one piece of the genome to another. And below here is the copy number alterations, the changes in the number of copies of an individual gene that they have. The point here is not to focus on the individual mutation types. The point here is instead to say, if you look across men, and the ones that I've highlighted here in the green box are those that have Gleason scores of 3+3 or 3+4. These should be men who have 6 or 7, relatively good prognosis. It should do very well. Well, we can see a huge amount of heterogeneity. Some of these men have over 3,000 mutations in their tumors, and some of these men have almost none. And we know that the number of mutations you have in a tumor is associated with worse prognosis. So the higher number of mutations you have, the worse the prognosis. And yet, within this broad group within this narrowly defined group of men who should all have good prognosis, we have massive differences in the numbers of mutations that we're actually seeing. Conversely, in men with Gleason score 4+3, which is a more aggressive form of cancer, if you don't know anything else, we see again this amount of heterogeneity within that group, but we see men who have very few mutations, whereas we see men over here with a better prognosis that have lots of mutations. And so while the trends are true, while these men actually do tend to have more mutations, again, like people tend to live to be about 82 years old, not every tumor is the same, and not every person lives to be 82 years old. Some of them are very high and some of them are very low. We have heterogeneity. And so the question is, how does that relate to outcome, and which of these mutations actually matter? And so I talked about the concept of driver mutations versus passenger mutations, and through a number of computational techniques, we could identify a set in these patients of about 40 mutations or sets of mutations that were quote unquote driver mutations. They seem to be responsible for promoting the growth of these cancers. So what you're looking at here is a list of all of the different mutations. Again, not important what the names of these genes or the features, just that we found 40 of them. They were of different classes, as represented here by the colors. The bars in the middle are representative of what's known as the hazard ratio. Essentially what that means is that if you have this mutation, for example, the hazard ratio reflects the likelihood that you will have aggressive disease. So if you have a hazard ratio over 1, it means you're more likely than, your chance is higher than the average. If you have a hazard ratio lower than 1, it means that your prognosis is actually better than average. And what's shown over here is just what's called the statistical significance of that. So of those 40 genes, 40 mutations that we found that were associated with, that we thought were drivers, 9 of them, and that's these 9 here, 1, 2, 3, 4, 5, 6, 7, 8, 9, were also associated with more or less aggressive disease. So this is what I was talking about earlier, where mutations can either promote disease growth, disease aggression, or both. Well, turns out these 9 seem to be doing both. And so when you plot this out, patients that had these 9 mutations, actually we used some computational techniques to reduce this down to the 6 most important features. But if you had those 6 features and you were quote unquote high for these features, the signature that was developed from these features, the men shown on the red bar, you can see that within 10 years, about 60% of those men had relapsed, compared to only about 20% of the men who were low. And in fact, if you looked at 6 years, almost none of the men who did not have these mutations actually had failure after surgery or radiation, whereas about 50% of the men who had the signature did fail. And so this signature allows us to better stratify men as to whether or not they're going to have aggressive disease or potentially metastatic disease after they've been treated. And remember, this is all on top of what we already know. So all of these people have the same Gleason scores, same PSA, same tumor size. So this is over and above what that information already gives you. And I apologize for the size of this. This is newer data where we show that it's not just the PSA recurrence that we see, but also in the metastases. So these men are actually getting metastases as well. If we go back to this concept of the kind of clinical course of the disease, in a good case scenario in localized disease, the person gets surgery, PSA drops back down, they might have a little blip, it gets treated with radiation, maybe sometimes hormones, PSA comes back down, and man comes in every 6 months for every year, and the PSA stays low. That's fantastic. And as we talked about, in a small number of cases, but a significant number of cases, that doesn't happen, and a man ultimately develops life-threatening metastatic prostate cancer. Well, over time, one of the insidious things about cancer is that mutations that cause cancer often do so by making it easier to get new mutations. So given enough time, mutations will accumulate, and they will accumulate at a faster rate. So you can actually mutate genes that are involved in preventing mutation itself. And so it's sort of this exponential growth. So what happens is mutations accumulate over time. So if you were to look at these tumors here in the localized setting that you got from the initial surgery versus tumors that you took out of a bone biopsy or a liver biopsy for a man with advanced metastatic prostate cancer, these cancers would have many, many more mutations than these, even in the same man, simply by the fact that the mutations accumulate over time. And so the question is, can you learn something about that? Can you learn something about the original disease by looking at what happens downstream? If you consider for a second the way cancer develops from a single cell, in this case, a normal prostate cell. And over time, mutations can occur in that cell, somatic mutations. So each of these lines represents mutations accumulating over time. So this is a normal prostate cell. This is a cancerous prostate cell that has mutations that have developed in this amount of time. And then over time, months and years, and of course, prostate cancer is a very slow growing, long development time disease, more mutations will develop. And so that cell will divide and will give rise to other cells that have new sets of mutations, and so on and so on and so forth. And you'll get these populations of cells, which are called clones, which share a sort of common backbone of mutations. This is the primary tumor. Not all of these cells are actually going to go on to lead to distantly metastatic, aggressive, advanced prostate cancer. Only a very small number of them will. So here's one cell that went on to seed some metastases, let's say in bone or liver. Here's another cell that did the same. Now, these two cells they both arose from this initial cell here, which itself arose from this initial cell, and so forth. So what we really want to do at the end of the day is not necessarily find out what are the differences in mutations in this arm of the tumor, because these didn't go on to seed metastases. We want to know what happened here. What happened here? What are the mutations that are present in this metastasis and this metastasis? Could we find those in the primary tumor? And if so, could we use that information to determine which primary tumors are likely to metastasize because they contain those mutations that are associated with metastasis? And can we do that before they metastasize, of course, so that we can tell a man, you have a high risk of metastasizing, as you have a small number of cells in your tumor that perhaps contain mutations that are associated with very aggressive disease. And so that's exactly what we did. And so this is work that's actually very recent work that we've done over the last 2 or 3 years now. And one of the great things about the drop in costs for some of these assays is that the amount of data that is now available to researchers around the world is huge, because so many groups can do this type of work. So what we did was we took a little under 1,300 localized cancers, and we had the genome sequences for all of those, and about 550 cases of metastatic aggressive prostate cancer. So for a total of just under 1,900 prostate cancers. It's a very large number for this type of a study. And we looked in those cancers and said, what are the drivers, right? What are the mutations that appear to be involved in actually causing those cancers? And so we talked about the 40 that we discovered in our localized cancers earlier. In addition to those 40, we also found 73 new ones in metastatic cancers. So for a total of 113 mutations across all of these 1,844 cancers that we thought were driving those cancers. And so our goal here was not just to find out what the drivers were, but let's ask the question, can we find which of these mutations are these mutations? How many of them are present in small amounts in the primary tumor? Not zero, but in small numbers of cells within the primary tumors. And learn about those mutations, and are those the ones that actually end up becoming enriched in the metastases, because they are the cells that ultimately go on to seed those metastases and cause the aggressive disease. And so that's exactly what we did. So this is some data that's actually just been accepted for publication, which we're very happy about. So this is a plot of our 113 driver mutations. Again, you don't need to be able to read this or know what the genes are. It's completely not besides the point at this point. What you should notice from this is that in the top panel, which is localized prostate cancer, this gene, which is called NKX31, was mutated in about 50% of all cancers that we looked at, 50% of those 1,289 cancers. And there were only 5 genes that were mutated in at least 20% of those localized cancers. The first 5 basically on this plot. When you compare that to metastatic prostate cancer, the most mutated gene in that disease state was a gene called AR, which is the androgen receptor, which is the target of androgen deprivation therapy, not surprisingly, is mutated in about 75% of metastatic prostate cancers. And there were 25 different mutations that were mutated in at least 20% of all of the patients that we looked at. So mutations are much more common, as we expected, in metastatic prostate cancer. There's a lot more of them, and they happen a lot more frequently. But do they happen more frequently than we'd expect by chance, because that's really what we're looking for. What we're really looking for is these mutations that happen not just because the cells are accumulating mutations over time, but because they've actually been selected based on their biology to go on and be more aggressive. So are they happening more than expected? And to do that, we need to go back to a little bit of probability and consider a coin. If you flip a coin 10 times, you'd expect 5 heads and 5 tails, 50% proportion for one and 50% proportion for the other. If you flip the coin 10 times and you saw 2 heads and 8 tails, could this be due to chance? Probably. I think we've all done that, where we flip coins and we get a streak of 5 or 6 heads in a row. It happens just by chance. But if we flip the coin 100,000 times and we saw 20,000 heads and 80,000 tails, could that be due to chance? Well, likely not, because we flipped the coin so many times. It's unlikely that that's actually a fair coin. It's likely to be biased towards tails. So it's due to chance. So what we really need to do is figure out which of these mutations is actually occurring at levels that are not due to chance. So we can't just flip a coin, unfortunately, because in some ways, while a mutation is like a coin toss, it's either there or it isn't. In some ways, it's not like a coin toss at all, because it's dependent on what's going on around in the cell. Long story short here is that we had to do 100,000 simulations, so computer simulations, on all 113 mutations for all 1,844 patients, which works out to 20,837,200,000 total simulations. And by that, I mean simulating the growth of a tumor to see whether or not that individual mutation would develop in that particular case. And we do that over 20 billion times. Now, that sounds like a lot, but fortunately, we can do that with the computer that sits under my desk in my office, which is not the 113th fastest computer in the world. And it takes about 20 minutes, which is really quite amazing when you think about it. You can do all of that data simulation and sort of grow, if you will, in a computer, a tumor 20 billion times, and it takes about 20 minutes to do that. And so what did we see? Well, this is the raw data. So each of these dots represents an individual mutation, and the horizontal axis is the frequency of that mutation in the aggressive metastatic cancers, and this axis is the proportion of that same mutation in the localized cancers. Here's the AR, as I said, present in almost 75% of metastatic cancers, and there you can see a number of others that are sort of occurring more frequently. Our question was whether or not they're occurring more frequently by chance. Well, again, long story short, about 37% of the mutations that we saw, those 113 mutations, were more prevalent than we would expect by chance. 24% were actually less prevalent than we were expected by chance. So they were actually being selected against. They were mutations that the cancer was saying, no, we don't want those mutations. They're actually preventing more aggressive cancer. And 39% of mutations were just as prevalent as expected. The ones that were more prevalent than expected are these ones here at the top of this curve. The bar here is basically the proportion of metastatic cancers that had this mutation. So the ones we were interested in here are the ones here that were sort of above this red line, which indicates whether or not we believe this statistically. So there were 42 mutations. That's 37% of 113. 42 of those mutations were present in higher than expected prevalence. They were more common in metastatic cancer than localized cancers. We then said, which of them are present in at least 5% of localized cancers? Because again, we don't want things that are in, if I go back, in these cells here, we want the ones that are in these smaller proportions, but not zero proportion. With 23 of those 42 were present at that level. Of those, we then asked which of them are associated with metastasis following surgery or radiotherapy. We found 4 mutations. 3 of them were mutations in genes that we already knew were involved in prostate cancer aggression. One of them, which is great because it means we're finding something useful, but it's a lot of work to find something that's already been found. The fourth we were really surprised by. And it was a gene called ZNRF3, which had never been looked at in prostate cancer, had never been observed to be involved in any differences in terms of whether patients did well or not. In fact, I think there were only one study of it in the context of prostate cancer at all. And so when we looked a little bit closer at this gene, we found that 25% of men who had metastasis after they were treated for localized disease. So these are men who go on to develop potentially life-threatening disease. 25% of those men have mutations in this gene, compared to about 5% of patients who do not metastasize, or at least did not metastasize during the time of our study. That's a lot. 25% is a lot when you compare it to other genes. And when you look at how patients actually, the outcomes for those patients, you can see that if you had this mutation, those are the blue curve here, at 10 years, again, about 40% of those men did not have a relapse. So 60% had a relapse, compared to about 40% who did not have the mutation. And again, it's the same when we're talking about metastasis-free survival. What was really interesting is this doesn't all occur in a vacuum, of course. We know other things about these cancers. If you put this mutation in the context of other mutations, here I'm showing a different gene mutated, but this is true of the other signatures that we had found in the past. If you have that plus those other mutations, almost all of those patients develop metastases within 10 years following treatment. And so we can say with very high certainty, above and beyond what Gleason score or PSA or any of those other things can tell us, we can say you are very likely, despite the way your tumor looks and your PSA values, you're very likely, much, much more likely than the average person to develop a life-threatening cancer. And so that's great. And you know, I think that is a worthwhile thing to do. But it's of no value unless we can somehow channel that into the clinic and develop ways of getting that information to men and their physicians, and making differences in terms of how men are treated in the clinic. And this is where it becomes a little bit more tricky, because, of course, as you know, to get these things into clinic requires a lot of validation, and it requires a lot of work to make sure they're working properly and they're accurate and all of those things. Our view is the best way to cure prostate cancers is not to treat metastatic disease better. It's to prevent men from developing metastatic disease in the first place. Frankly, we'd love to be able to prevent men from getting prostate cancer in the first place. But if allowing for that happening, a better approach is to keep men in a localized state, cure them while the disease is localized and not metastatic. And our goal is to take, if you think of the disease along a continuum from really true indolent low-risk disease all the way up to castrate-resistant, I hate the term castrate-resistant, but castrate-resistant disease, very aggressive disease. What we really want to do is more accurately place men into these bins so that they can receive the treatment modality that's more associated with the true aggression of their disease, not the average across populations, but actually the true aggression of the disease that we know from the actual biology, the actual DNA that's in their individual tumor. So we identify who will progress, treat those men more aggressively, and perhaps more controversially, identify men who will not progress and treat them potentially less aggressively and spare them side effects that they don't need to have. So how does that look? Well, one potential way forward to that is, again, if we go back to the blue and red men analogy, right now, we're colorblind to this. All men presenting with, let's say, intermediate risk prostate cancer look the same. What we want to be able to do is tease out who the blue men are and who the red men are. And we want to be able to do that accurately. So how do we do that? One idea is that we take all patients, and as a normal standard of care, we offer them a test. So it could be, for example, testing for this mutation that I showed just a minute ago, ZNRF3. Could be looking at the 5 or 6 feature signature that I showed you earlier in the talk. There's a lot of potential candidates here. But the idea is, and maybe it's more than one, the idea is we develop a test. If you test negative, that is, your tumor does not contain those mutations that make up that aggressive signature, then you would be treated as you would be today with standard of care. And generally speaking, that's again precision radiotherapy or surgery alone. And the thinking is that will sort out all the men, all the blue men, with very, very few red men getting through. Right now, it's about 30%. We want to get that down to, well, hopefully nobody. If, on the other hand, the test comes back positive, you have these mutations, what does that tell us? Well, in the context of a clinical trial, what would happen is a man would be randomized based on that test to either receive the standard of care or to receive standard of care plus something else. So that could be something as simple as a short course of androgen deprivation, or could be targeted agents based on things that we are learning about the biology of prostate cancer cells that have these mutations. It could be surgery followed by a short course of radiation. There's a lot of different ways that treatments could be modified to suit the actual aggression. And the idea would be, past the clinical trial stage, is that everyone who tested negative gets the standard of care, everyone who tested positive gets the standard of care, plus this intensification, and that we would be able to then refine our analyses so that no blue men, or very, very few blue men, are being overtreated for disease that's not going to threaten their life, but all the red men are getting the actual, the level and intensity of treatment that they actually need to prevent them from developing the metastases that would have happened anyway had they not undergone this more intense treatment. Our approach to that has been to create what's called a panel, a sequencing panel, and this is, I just have a couple slides left here, and I'll just talk about some of the things that are sort of ongoing in our groups, is to design this panel where we take that 3 billion letter genome, which is a lot. And even though we can sequence the whole genome in 7 days, the amount of data and the computing power that we need to do this is vastly, vastly larger than most institutions, certainly non-academic institutions, community physicians and so forth, can actually handle. We distill that down to about 1% or slightly less than 1% of that whole genome into the bits of the genome that actually mean something to a prostate cancer patient because they're informative. They tell us things about what that disease, the biology of that disease, the clinical course of that disease. And we offer a man a test whereby the DNA of that tumor is tested each and every time for each and every patient that comes into the clinic. We can turn this around in, let's say, a week. So at your next follow-up appointment, the physician is able to say, good news, your tumor tested negative for our signatures, and therefore your chance of developing metastatic disease isn't the 10% that the population has. That, again, that average across the population, it's not 10%, it's less than 1%, or even better. Conversely, you had 4 of the 6 mutations that are associated with aggressive disease. Consequently, you're not actually a low-risk disease. You're not really a good candidate for active surveillance, for example. We really do need to treat that tumor because it's not a 1% chance of relapse. It's actually 15 or 20%, and that changes the calculus. And so that is the type of approach that where we can take these things and move them into the clinic by translating them into tests that are cheap. And in Canada, of course, we have single-payer healthcare system whereby things have to be, and even in places like the United States with multiple payers, we still have this issue of the economics of these things and how much do they need to cost for governments to reimburse. The good news is that these tests can be done for $300, which is in line with a lot of what governments are already funding in this space. And so this is the type of approach that we're taking. We're working with our partners here in Ontario, with Cancer Care Ontario, but also with other organizations across the country to sort of look at the feasibility of this. And we know we can do it from the standpoint of the data collection and data analysis. It's really about how do we actually move this into the clinic and drive this into a test that can make differences. This is my last content slide. I will simply say that one of the unfortunate things about this process is that it is not linear. I sort of liken it to climbing Everest. You arrive at base camp, you hike up to camp 1, and then you hike back down, and then you go up to camp 2 and you hike back down, and you get acclimatized, and it's an up and back process before on that final day you can go for the summit push. Biomarker development and implementation is very much like that. We've done the discovery. That's what I've shown you today. We've done a lot of the retrospective validation where we looked in other people's data sets around the world. And we have groups now of 2,000-3,000 patients where we validated a lot of these things. We're now doing the assay designs where we can actually design these chips to put them into clinic. But what will happen is next week, there'll be papers that come out that identify new genes and new mutations, perhaps, that we missed for one reason or another, or we'll find that a gene interacts with another one, or these BRCA mutations. And I haven't talked about the familial side of this thing, but they can interact differently with some of these mutations, and they change the way these tests look. And so we have to perhaps go back and relook at our data and iterate in these things. But ultimately, this is where we're moving toward. We're moving toward these types of tests moving into the clinic in ways that can much better and tell an individual man, you have more or less aggressive disease. We're not treating you as an average. We're not treating you as a population. We're treating you as an individual because we know now what the unique biology of your tumor is. I'll stop there. As I said, I'll be happy to take any questions. This is a small proportion of the people who worked on this study over the last really 15 years, I guess it is now, 2011 when we launched this study, 10 years, and then a little bit before that, so 10 to 15 years. These are all the people that have worked really from around the world. But more than any of these people, I really want to thank, with respect to the CPC-GENE program, the 573 families and counting who have trusted us with their tissues and private medical information during the most difficult period of their lives. And none of this would be possible without them and the people around this call right now. And so thank you very much. As I said, I'd be happy to take any questions you have. Thank you very much for the invitation.
Thank you so much. We did get a couple of questions here in the chat that I'll ask first.
Sure.
Okay, so at what age does this start showing up in the DNA?
Right. So there is a little bit of data on this. Younger men have a different form of cancer than older men. I think we know that now. So there's some interesting data that has come out of some of our colleagues in Germany where they've looked at DNA mutations in men under 50 who had prostate cancer and compared that to men over 50. And they can see very distinct differences in the types of mutations that happen. Now, what causes that to happen, what causes the mutations to be different is not clear at this point, but it is clear that in general, prostate cancer in men over 50 is associated with a fairly well-established set of mutations. And so they happen, it's hard to say what age, but probably somewhere around in the 40s and the 50s where they start to build up these mutations. And it's not an overnight thing. It's not like you get a mutation and tomorrow there's cancer. Prostate cancer takes years, decades to grow before it even becomes clinically noticeable. So it's probably in that sort of state, in the sort of 40 to 50-year-old range. Conversely, men who are being diagnosed with prostate cancer under 50 years of age, those mutations are happening much earlier. And in fact, they may even be associated with mutations that were there at birth. Not necessarily BRCA1, BRCA2 mutations, but mutations in other genes that are maybe a little bit more subtle in how they work, that make it slightly more likely that certain mutations will accumulate. And so those mutations in men who are, the 45-year-old who has prostate cancer, those mutations are happening probably in the man's 20s. We don't know that for sure because we don't have enough of them studied at this point, and we don't have the data from that early on. But my guess is that you'd be looking at mutations sort of starting much earlier in life. I hope that answers your question.
Thank you. Next one is the test easily available to the cancer survivors to ascertain the active genome that may cause metastasis, the blue men and red men? What way could it be treated before it mutates?
Right. So, so I want to reiterate, the tests right now are not part of the clinic. There is no clinical test at the moment. Those clinical tests are in development right now, and there are teams both here in Toronto as well as our collaborators and colleagues in the United States who are working actively on this right now. So our goal would be to have this be part of the routine standard of care, whereby if you come in to a major cancer center, at least at first in a major cancer center, and we'd hope to have this be adopted in other settings as well. But that if you come in, you're getting this test. So it's just like everybody gets a PSA test. Everybody gets a biopsy. We're seeing now more and more, again, particularly in larger centers, people are getting MRI. Our hope would be that this is another piece of information that can be added to the calculus that goes into defining the aggression of a man's disease. In what way could be treated before mutations take place? That's a very interesting question, and it is an active area of research. Prostate cancer prevention is, I think, more what you're speaking about here in terms of, are there ways that we can prevent those mutations from occurring in the first place? The answer is we don't know, unfortunately. It's not like lung cancer where we sort of know if you stop smoking, your risk of getting lung cancer goes down enormously in the first year and continues for many years after that. Unfortunately, in prostate cancer, the actual epidemiology of what the mutations are or what's causing them isn't particularly well established. There are certainly clinical trials that have been done around dietary changes. There are other environmental factors that might be involved. And there are studies ongoing here in Toronto at the Ontario Institute for Cancer Research that are directly leveraging large data sets of 300, 400,000 Canadians to look exactly at that question. So what are the environmental issues, and if there are clear contaminants that result in certain mutations, could we avoid those things? There has been historically some chemo prevention strategies for prostate cancer, and things like finasteride and dutasteride, or these 5 alpha reductase inhibitors that like Rogaine is a 5-ARI inhibitor, and actually is a drug that was first developed for prevention of prostate cancer. That's a little bit dodgy now because there's some data suggesting that they may actually, while they reduce the risk of developing these mutations, if the mutations do happen, the disease may be more aggressive. It's not quite clear. But unfortunately, the answer is there's not a whole lot we can do once the mutations take place. Which is why I think a lot of the focus for us and others has been to sort of say, yeah, let's diagnose the disease much earlier. Let's use PSA and other tools to diagnose disease when it's very early. And if we don't do that, let's also, or even if we do that, let's use these tests to really figure out who needs to be treated more aggressively. Because, as I said at the onset, anybody who's in the clinic today with clinically localized prostate cancer should be curable because the disease has not spread. And yet we know that 20 to 30% of that times that's not going to be true, depending on the risk. So I hope that helps to address your question. Thank you for the question.
Okay, next question. Are these tests only done on extracted prostate tissue rather than the bloodstream? If so, is prostate tissue available following, or would my prostate tissue be available following my prostatectomy 5 months ago?
Right, so they're actually done on both. So we need bloodstream DNA because we need it usually to establish what your baseline is. So we use bloodstream DNA sort of as, this is what you were born with. That's not technically true, but it's because there may be things that happen in your bone marrow or whatever during life, and it influences the genome in your bloodstream. But we can at least say that the tumor has sort of been built on that backbone, let's say. So we need that to sort of say, here's what man X looks like at time 0. The tissue is what we really want and is actually now being pushed into things where we don't even need the tissue, where we can actually extract DNA from the tumor in the bloodstream, which would be even better, because then we wouldn't even need to have the prostatectomy. So for research purposes, we prefer to have the prostatectomy because we can then work with the large amount of tissue that comes out, and we can pick and choose the regions that we want. We've done also work on biopsies because we can get a fairly good amount of material out of biopsies. The sort of black humor joke that we tell ourselves is, it's no good to tell the man, good news, we looked at your prostatectomy, you have an excellent mutational profile, and you didn't need to have a prostatectomy after all. That's not what we're shooting for. We need to do that before. We need to be able to say, you don't need a prostatectomy. And so ultimately, we need to be working on biopsies. We need to be working on, even better than biopsies, because of course we know that biopsies have complications. We need to be working on material that we can get from the blood, tumor material that we can get from the blood. So this is quite an active area of research in aggressive, advanced prostate cancer, where there's, because of the amount of tumor in the body, that tumor is sloughing off DNA into the bloodstream all the time, and you can actually extract that and sequence it and see what's going on in the original tumor. In localized cancer, it's a little bit harder because there isn't so much tumor shedding off DNA, but there are some technologies, again, developed here in Toronto at Princess Margaret, to actually look at some of these mutations in blood. But certainly, if you had a prostatectomy at a large institution, almost every single one of them that I'm aware of would have had some kind of biobanking program whereby the prostatectomy is sectioned. Part of it goes off to the pathologist to look at it, and then a part of it goes into a liquid nitrogen tank to be stored for research purposes. So we absolutely use that type of tissue. It works very, very well, and is the ideal material to work for research purposes. But on a clinical test, we need to move away from, we need to be able to do this on, at bare minimum, biopsies.
Okay, next one. It appears that you know the medical history of your parents, or perhaps other family members, is important in understanding the risk of cancer. Should we be documenting this information when consulting with our medical team?
Yes, absolutely. So the thing about family history is that the BRCA1 mutations and BRCA2 mutations, while they increase risk of developing breast, ovarian, a number of other, prostate, pancreatic cancer, they do so, they increase the risk by, let's say, 5 to 10 times. But they are actually quite rare, those types of mutations. So a very small number of cases can actually be attributed to those types of mutations. Much more common is mutations that are, again, inherited from the parents, but that increase the risk by 0.01%. And so for an individual person, they may never see that. But over populations of 10 or hundreds of millions of people, you will see that. And so you need large groups of individuals, both with and without cancer, to actually tease some of those things out. That having been said, absolutely family history, particularly first-degree relative family history, is absolutely critical that your physician knows that information. And it's not just prostate cancers. It's first-degree relatives, so fathers, mothers, sisters, brothers, children, with breast cancer, ovarian cancer, pancreatic cancer, particularly male breast cancer, bilateral cancer. So for example, women getting breast cancer in both breasts, that's very, very often associated with germline mutations that are associated with higher risk. And I say this not just because it will benefit you as an individual to know that information, but for two reasons. One is it will help, if you knew you were a carrier of a mutation, it could potentially help your children have that information and make decisions for their own health care or the care of their children. But also there are now therapies that are guided towards men with specific sets of mutations in their germline. For example, men with BRCA2 mutations are now, in the United States at least, approved to be treated with a drug called olaparib, which is a class of drugs called a PARP inhibitor, which actually works specifically on tumors where there is a mutant copy of BRCA2. So it's absolutely both from a preventive and how do we deal with potential things coming down the road issue. We need to know that information, but also it may actually influence your care at some point down the road. So absolutely should have that information if it's available.
Awesome, thank you. So that's it for questions in the chat. Does anybody else have anything?
A supplementary question of the question which I had already sent.
Mm-hmm.
All right, and that was related to, is the test easily available? And then you responded it so well. I understood it quite well. Now, my question is that doctors, since we don't have a solid, candid research, so doctors rely on a broader spectrum of treatment. Like, for example, if a doctor suspects, like in my case, it's intermediate, high risk in terms of 4+3 Gleason level. So the doctor thought, why, Jim, you are really tolerating this quite well, your hormone therapy. Can you also have Zytiga, or what you call in generic name of Zytiga is aberration acetate. Yeah. So would you like to have that? I said, well, it's your choice. But then I consulted one or two more doctors, and then they said there's no requirement. So my question is that how the patient should really take a final decision once, in the absence of a candid research, whether this kind of a treatment is going to stop metastasis or it's going to really create a side effects or more harm than the benefits. Yes.
Yeah. So you can imagine this is a complex question. And so the answer is that it is unfortunate that the evidence for the efficacy of certain treatments, for example, you mentioned abiraterone, Zytiga, the utility of that drug or lots of other drugs like it in specific scenarios as compared to the one you mentioned, people are not applying evidence equally in different centers. So it is a difficult decision to make. I can't speak specifically to the evidence with respect to Zytiga. I'm not an expert in, well, first of all, I'm not a physician, and I'm not an expert in the context of advanced prostate cancer. That having been said, my best thinking there is that there are organizations like Movember, for example, Canadian Cancer Society, Prostate Cancer Canada is now associated with Canadian Cancer Society, that very often publish reviews on these types of questions, exactly these types of questions, and will actually go through the scientific evidence and clinical evidence to support one way or another. There is also a resource in the United States, which is part of the National Cancer Institute in the United States, called the PDQ, so Papa Delta Quebec, which has literature on the internet. If you look this up, there is one half which is geared towards physicians, so it's more of a technical literature review, but a part of that is also to develop a patient-facing resource. And a lot of times it will take the scientific evidence for those things and actually distill it down to, here's what the recommendation is. And this is all through the National Institutes of Health in the United States and the National Cancer Institute. So you can look that up as well, and it may have some, I don't know for sure, but it may have some information about that specific question.
Thank you.
Thank you. Anyone else?
Oh, yes, I have a question. I think it's covered already, Dr. Fraser. Thank you. It's just been an absolutely great, articulate, and understandable presentation. So that's very nice. Thank you. But just to clarify what you've said, so do I understand then that my daughter, for example, has an increased risk of breast cancer, just as my sons have an increased risk of prostate cancer because I have it?
Right. So there's two ways to look at it. So let's assume we knew nothing about your genome, okay? And I don't. This is the first time we've met. If you had let's say a brother that had prostate cancer, or if your father had prostate cancer, or your sister had breast cancer, so forth, then not knowing anything else about your genetics, the risk for you would be higher. And consequently, because you have prostate cancer or had prostate cancer, your sons would have a higher risk without knowing anything. Now, if we did the test and we found that you did not carry a BRCA1, BRCA2 mutation, you didn't have mutations in other genes that we know are associated with increased risk, then your son's risk would then not be as high anymore because we have more information. So we've now taken, right? So we've taken that, let's say it went up by 5 times because of the family link. But now we actually have the genetic information. Well, we can now discount a family link because there's nothing that we can see, right? So having that information changes the calculus of whether or not there is an increased risk. So I guess the point is that there is familial linkage where you sort of observe a lot of associated cancers, but prostate cancer and breast cancer are common even without any family link. So it could just be by chance. So you really have to do the testing to then say, is there really a genetic link here? And if there is, then you can deal with that as an absolute increased risk rather than a sort of assumed increased risk because of the family linkages.
Well, thank you. So it does apply to my daughter, though, the same way as this general risk applies to my sons. Because she's a daughter, her risk of breast cancer is is higher without knowing any more information.
That's right.
Yeah. Thank you.
That's right.
Perfect. Thank you.
Anyone else have any questions? I have one. Well,
Okay.
So we've had companies like Invitae and stuff reach out to us about their genetic testing. What would be the benefit of doing that?
Well, again, it comes down to one of two things. The first is if you were diagnosed with prostate cancer, where you were treated for your prostate cancer successfully, or maybe you had a PSA relapse or something, it could be of clinical value. My view, and again, I reiterate, I'm not a physician, is unless there is a clinical value, that is, unless you can change how you're going to, if you're going to give a treatment or not give a treatment or change a treatment or change a dose, a test for the sake of doing a test is of no value. It's only of value when you can implement it in some way. So there's 2 ways you can implement it. Number one is, as I said, knowing that you had a specific mutation could, at some point in the future, change the clinical management. Because if you were to go on to develop metastases, there are drugs available today that are approved for men with specific mutations in specific genes, and they work very well. So knowing that is important. And as I said, the other thing is that test has value because it changes the information for children, potentially, and their children, because now you know, yes, there's a mutation. Okay, therefore, I should have my children be tested as well, perhaps, if that's a decision they make, and they can have that information. And I should point out, of course, the way it works is that if you have cancer and you have a mutation in one of these genes, your children may not. It's not necessarily so that you will pass along the mutation. You may or you may not. So having that information is just another piece of the puzzle to assess risk, right? The background risk for prostate cancer is about 1 in 9 Canadian men in their lifetime. If you were a BRCA carrier, it would be something like 1 in 2. So even then, it's not 100%. You're not for sure going to get it, but it is another piece of information. So there's lots of companies that do it. If you are so inclined, talk to your physician, and next time you have an appointment, and say, I've heard of these types of tests. Is there reason to suspect that there might be a familial link? Most large institutions have genetic counselors on staff, or can consult to genetic counselors, and they can help you through that process of assessing risk. And under certain scenarios, that testing is actually covered by provincial health plans. Here in Ontario, there are certain guidelines where if you have more than one first-degree relative, you can be tested. Sometimes it's not, in which case it's out of pocket. It's usually a couple of $100 to do that type of testing. But there are resources available to actually find out that information, and to help, not just find out, but to help you make sense of it, and then to help you make decisions based on that information.
Perfect. Thank you so much. Anyone else have any questions? All right, I think that's it. Thank you again so much. I'm getting lots of positive feedback in the chat here about how awesome your presentation was. So thank you.
It was my pleasure, and thank you again to everyone for the invitation. And yeah, just a pleasure, and best to everybody here.
Thank you.
Thank you.